Good day to everyone joining us, and welcome to today's Xtalks webinar. Today's talk is entitled How to Make Adaptive Dose-Finding Trials Easy for the Clinician. My name is Ryan Muse, and I'll be your Xtalks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, please contact me at any time by sending a message using this same chat panel. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming on xtalks.com. At this point, I'd like to thank Atlantic Research Group, ARG, who developed the content for this presentation. ARG is an oncology, immunology, rare and neurodegenerative disease-focused contract research organization that provides comprehensive clinical program development services ranging from pre-launch consulting to commercialization, achieving over 50 market authorizations. Founded in 2004 with the vision that every project should be highly individualized, ARG has experienced consistent growth across the globe, expanding their reach to include drug and device strategic consulting, clinical trial management services, and clinical data and analytics solutions. Now, I would like to introduce our speakers for today's event. Dr. Friend has more than 17 years of global drug development and medical affairs expertise in hematology and oncology, as well as a variety of other therapeutic indications. He is currently the Chief Medical Officer of Cellectar Biosciences, leading its research and development, clinical, medical affairs, and regulatory affairs divisions. He completed his postdoctorate residency program in family medicine and subsequently served as clinical director and faculty attending physician at Cabarrus Family Medicine Residency Program in North Carolina. Dr. Schnipper is a widely recognized expert in innovative design and management of clinical trials in several indications, particularly in oncology. He has established a reputation for being able to determine quickly and cost-effectively as to whether a drug is safe and effective. Currently the head of clinical development at X37, Dr. Schnipper has played key roles in drug development from IND to NDA in diverse organizations. He practiced medical oncology in both academic and private practice settings before beginning his pharmaceutical career with Hoffmann La Roche. Dr. Wisniewski has over 30 years of experience as a statistician in clinical research. He is currently the VP of Biostatistics and Data Informatics at ARG. Previously, he held similar positions at Covance, formerly Chiltern, PPD, and GSK, where he has built and led international teams of statisticians. He has designed and led projects in many therapeutic areas, including oncology, rare diseases, infectious diseases, respiratory, GI, and metabolic diseases. He has worked on several regulatory submissions and has led teams that have worked on others. So what is this webinar about? If it could be summed up in one sentence, it is about using adaptive design techniques to reach the MTD, maximum tolerated dose, in early phase trials, which could result in clear measurable benefits to both patients and sponsors without adding complexity and anxiety. And so now, without further ado, I would like to hand the mic over to our first speaker, Dr. Schnipper. You may begin when you're ready.
Thank you. So today we're going to talk about adaptive clinical trials and the idea being of how you can add precision to your program without adding complexity. I, for one, have been doing clinical trials for a number of years now, and I've been totally comfortable using a more standard 3+3 design. It's always gotten it to work for me. When I read about adaptive designs, the first thing I'm confronted with is complicated equations and statistical methodologies. But just like I don't need to be a software engineer to be able to send an email, I actually don't need to understand all that intricate math in order to be able to utilize these designs to benefit a clinical program. So I think one of the things that we would like to focus on today is the fact that the complexity really doesn't have to be a barrier to using these designs. So the first thing we'd like to start off with on the next slide is some of the advantages of these kinds of programs.
Thanks, Ed. That's my cue, isn't it? Appreciate everybody for attending. So as mentioned, we're focusing our discussion today on oncology drug development, but much of what we're talking about can also be applied to pretty much any other therapeutic indication. Slide starts out with the question: what are the benefits for adopting adaptive designs? Maybe the more appropriate question is, who actually benefits from these adaptive designs? And I like to keep things real simple. So in reverse order of importance, and making sure everybody's awake and have their coffee, there are three key groups that really benefit. So sponsors, investigators, and of course, most importantly, patients. So who wouldn't really want to arrive safely at a go, no go decision quicker? Enrolling fewer patients could mean getting to that go, no go decision earlier, resulting in potentially significant cost savings that could be shifted to your phase 2 program, or as frequently is the case in drug development, utilizing those savings for unplanned cost change orders. Clinical researchers, investigators are keenly interested in novel study designs or statistical models, albeit many of us have a little bit of knowledge that makes us a bit dangerous, but in the end, we have to really rely and trust our statistical colleagues. Clinicians always have the best interests of their patients in mind, and exposing many of them to novel drugs that are clearly subtherapeutic is, can I say, maybe a little morally challenging, to say the least. Lastly, but most important, the patient volunteers. I can't imagine where the cancer treatment paradigm would be without those hundreds of thousands of cancer patients who participate in research. I do think that there is some expectations or hope across patients, families, caregivers, significant others of that potential clinical benefit, specifically, especially in oncology, response rates, overall survival or progression-free survival, beyond just the true altruism for trial participation. As such, safely limiting the exposure numbers of patients to low, what we know is non-efficacious doses in a methodical and safe manner is a win-win-win for all. Ed has already nicely laid out the goals for today. In addition, I'd like to throw out that it's not our goal to throw the 3x3 traditional design out with the bathwater, but it's to create some additional awareness around other methodologies, such that when you're planning your next early phase clinical trial, that you'll at least feel comfortable asking your statistical colleague if they've considered alternative methodologies. The next slide, Ryan, is a quick audience poll. So let me have you take it from there.
Yes, thank you very much. So we have a poll question for audience members to interact with. This should be appearing on everyone's screen right now, and you can interact with it by selecting on any of the answers you see in front of you and then clicking submit. The question we have for you is, why aren't more trials using adaptive designs now? Your answer options are, I don't know anything about adaptive techniques. I don't want to make protocols more complicated. I am apprehensive to try new techniques. I wonder if the effort does not yield large enough rewards. Or I don't want to make regulators figure out a new model. Your question again is, why aren't more trials using adaptive designs now? We'll give you a few seconds more to consider your answer, submit an answer for us. We appreciate you taking the time to participate. And it looks like most of you have submitted an answer. Thank you very much. Let's take a look at the results that have come in. It looks like 37% saying, I don't know anything about the adaptive techniques. 26% for, I wonder if the effort does not yield enough results. 14%, I don't want to make protocols more complicated. And then 11% for both, I'm apprehensive. And also, you don't want to make regulators figure out a new model. So once again, thank you for your participation. Dr. Schnipper, it's back to you.
Thank you. So what are the first steps? Well, I think we've already established one first step is to consider adaptive designs in the program that you're working on. Now, as was already said, this is no reason to throw out more traditional designs, but just to consider whether or not adaptive designs would be helpful to your program. And I can imagine, for example, certain situations where it would not be helpful, namely where perhaps I'm testing a drug over a very limited dose range because of issues of pharmacodynamics or pharmacokinetics, and perhaps these designs would not be helpful. But once I do decide that I want to explore using more adaptive designs, how do I actually deal with that? Clearly, getting a statistician involved at the very beginning would be important, particularly one who's knowledgeable in these designs and can help me understand how I can use these designs to most efficiently get to my desired dose for a phase 2 program with the least number of patients being treated at toxic doses or ineffective doses. In terms of the role of the clinician, it turns out that what a clinician actually has to provide to the statistician really isn't that much different than what they would need to provide to design a more traditional clinical trial. So first thing I would need to establish is what is my starting dose? And this is clearly something I would have to decide upon regardless of the trial design, a dose that I would derive from my preclinical tox program and my knowledge of that particular drug. And the other key parameter that I would have to provide is what's the highest rate of toxicity that I would accept for this drug? What percent toxicity, percent adverse events would I, at which I would stop dose escalating and consider that dose level the highest level of dose limiting toxicities that I would accept. And that's basically not much different than I would have a knowledge of if I was doing a more traditional clinical design. So with that in mind, let's move on to some of the more detailed attributes of exactly how these trials work.
Thank you, Ed. So following with Dr. Schnipper's comments about designing a trial, the objective in dose finding is basically the application of a version of the Goldilocks rule. You know, is the current dose too low? Is it too high? Or is it just right? The 3+3 design does this by looking at cohorts of size 3 and expanding to 6 if necessary. In some ways, it's a blunt instrument, but we'll show that a little later on another slide. Over the last 20 years, there have been many designs studied and evaluated that work towards this Goldilocks rule, and there are many publications that show the advantages of these newer designs over the 3+3. One prominent disadvantage of the 3+3 is that it underestimates the MTD in a majority of experiments. So if you have an experiment that can get closer to the true MTD, that certainly is a benefit for everyone. And although these newer designs are constructed so that outcome is to get as close to the true MTD as possible, the 3+3 remains very popular. As we saw in the poll earlier, you know, folks are reluctant because they don't understand the techniques, or maybe they don't want to make things more complicated. Well, that's what we're trying to do here today, is to show you that it doesn't have to be complicated. And a lot of this complication comes from the use of Bayesian statistics, which is a word that scares a lot of people. But these designs are simple to implement. But let's talk a little bit about Bayesian statistics first. Next slide, please, Ryan. So what is Bayesian versus frequentist? You know, statistical inference using Bayesian methods requires a different perspective from the frequentist world. The clinical researcher knows what a good outcome looks like. In frequentist statistics, the experiment is designed to find what the probability of this good outcome or a more extreme outcome would be over the course of many trials. In Bayesian statistics, the different perspective from what I just said comes from the probability expresses the belief that the clinical researcher has in the outcome. So the clinical research starts with some belief that that they will see a particular outcome or a good event, and then they have an idea of how likely that is to happen. The nice thing is that as you collect data, that belief can be updated. And one advantage is that this updating can be done as single observations are collected, and there are no penalties for adding additional patients to the analysis. The methods require prior knowledge, which is something that's different from frequentist statistics. And basically how it works is one has an estimate, collect data, and then you update your estimates based on the data. So to get started, for that original estimate, you can use the information that you already have, or you could use a method which is called in the Bayesian world a non-informative prior, which can loosely be translated as starting with no information and letting the techniques guide you. So what I'd like to do next is take a look at some of the commonly proposed designs, and I'll try to show you that they're as easy to use as the 3+3. But I do want to stress that it's important to consult with a statistician to help with the material. So looking at the 3+3 design, what do you need to get started? Well, there's no input required, no software needed, and there's no technical expertise required to apply the design. That's pretty simple. So what you do is count the dose-limiting toxicities within each dose cohort. Count the number of patients with dose-limiting toxicities of a cohort of 3 or 6. And then the rules are very clear. So we can look at a table of what the outcomes really show. If I can see the next slide, please, Ryan. And this is a schematic. It converts the rule of the 3+3 into a decision table. So in your cohort size of 3, if there are no patients with a DLT, well, certainly you escalate. And put an X over in the 6 box, because if there are no patients with a DLT after 3, you're never going to enroll 3 more. If you observe 1 patient in that cohort of 3, well, then the rule is to enroll 3 more patients. And if there's only 1 after you've enrolled 3 more patients out of a total of 6, well, then you can escalate. Now, the blunt instrument that I was talking about is that in your first cohort, if you see 2 out of 3, then the rules for the 3+3 are to stop and use the previous lower dose. And if you've gone into 6 patients, suppose you have 1 patient with a DLT in your first 3, and then you enroll 3 more. If you see another patient with a DLT with a total of 2 out of 6, the rule is the same. You terminate and use the previous lower dose. So this is kind of a penalty that you pay in the 3+3, that when you see 2 patients with a DLT, pretty much done. So there are other designs. Some of the more recent ones are shown on the next slide. There's a category of techniques which I've labeled as the toxic probability interval methods. TPI was first published in the mid-2000s, and since then there's been a modified TPI, and now a second version of the TPI, of the modified TPI. So what do you need to get started? This again refers back to what Dr. Schnipper was saying, is that you need a target toxicity rate. And for example, here we'll say that that value is 0.3, which means you would tolerate about 30% of the patients receiving the product to experience a DLT, but no more. Now, for these interval methods, you need to specify an interval or sort of a tolerance that works around them. Now, for example, I'll just put here, say 0.25 to 0.35, although it's a little different in the TPI method. And you're required to use some special software to do it, but there are many versions of it that are available on the web. They're free, and you can do it right on your desktop. How it works is you count the patients with the DLT. Your intervals are defined by the math and the process, and it generates a probability that the current dose under investigation is too low, too high, or just right. And when you're in that middle group, you can add additional data and continue to research that dose. So you need a statistician basically for the setup. The clinician can review the results. Decisions are easy to make, and the table on the next slide shows what this looks like, similar to what we saw on the 3+3. And as you can see, you can evaluate this after single patients. One thing I want to mention is that if a DLT is observed on the first patient of a cohort, it's recommended that you expand the cohort to 3 patients before you take any action to escalate or de-escalate. Certainly, if the second patient in that if you decide to expand your cohort with 3 patients and the second patient experiences a DLT, you probably will want to de-escalate. But you can see here, even with 1 out of 4, you get a chance to escalate. Whereas in the 3+3, you would be waiting until that 6th patient was finished before you make a decision to escalate to the next higher dose. There's another method, a sort of a modification of the 3+3, which is on the next slide, and it's called the interval 3+3. So what do you need to get started? Again, it's the same information: your target toxicity rate, some value of what you would call an equivalence interval. You calculate the toxicity rate for a given dose, and I promise these are the only 2 equations in this entire presentation. And you know, X here is the number of patients with a toxicity, and N is the total number of patients evaluated at that point up to that time. So you calculate what I've labeled as a low toxicity rate for a given dose, which is basically X minus 1 divided by N. And the rules are based on the 2 toxicity rates and where they are in relation to the equivalence interval. And so we can construct a table just like we saw, which is shown on the next slide. And you can see the difference between this and the 3+3 is that 1 patient with a toxicity out of 1, you would stay at the same dose until you've enrolled at least 4 patients and see only 1 toxicity. Then on your 5th, you would have an opportunity to escalate. So again, what we're seeing here is that you don't have to go all the way out to 6 patients when you've seen a patient with a toxicity in your first cohort of 3. And then the final method that I'd like to talk about on the next slide is a Bayesian optimal interval design. And again, what do you need to get started? You can start with the same toxicity rate. Now you can manipulate that rate if there are a lot of products in the market and you're bringing, say, maybe a 3rd or a 4th, then maybe you want to reduce that rate. Maybe a safer product would be more desirable. So what this technique does is it computes an escalation and a de-escalation boundary. The special software is required for it again, but as I said before, it's free and available on the web. There are many places you can find it. And so how it works is that you calculate the DLT rate for a given dose. It can be calculated for any cohort size. And then you compare it to the escalation and de-escalation boundary values. You can add additional data as it's available. And again, you pay no penalty to analyze the cohort with new information. As I said before, the statistician is probably needed for the setup, and the clinician can review the results. We've got a similar decision table on the next slide that can show you again how easy it is to make the decisions here. So for a target toxicity rate of 0.3, the escalation limit would be 0.236, the de-escalation limit is 0.358. So what that means is if you observe a DLT rate of 0.5, well, since that's greater than 0.358, you would de-escalate. And likewise, if you see a DLT rate of 0, it's lower than 0.236, so certainly you would want to escalate. Now again, the recommendation is if you see a DLT on your first patient, it's advisable to enroll at least 3 patients before you decide to make a change. And so again, here you can, if you observe 1 toxicity or 1 patient with a DLT, you can escalate after you see 5 patients, 1 out of 5 patients. And so clearly, again, you don't have to wait for that 6th patient to escalate. One feature that's nice about this is if this and with the MTPI and with the 3+3, I'm sorry, the I3+3, if you have no patients with a DLT, if your first patient doesn't experience a DLT, these methods allow you to escalate. And you may want to do that because, number one, that dose may be, the researchers may think that dose is not effective. It's in the protocol, as part of a dose ranging paradigm. But if there's no DLTs, there's no reason to continue to expose patients to a dose that may not be effective. And so with that, I believe we have another poll question. Is that correct?
We do indeed. Thank you. So another poll question we have for everyone right now should be, as the previous one appearing on your screen at this moment, select any of the 4 answers in front of you and then click Submit. The question this time around is, after attending this presentation, how much more are you inclined to use these techniques? Your answer options are: I feel exactly the same as I did before the webinar. I still have some questions, but I am willing to consider it. I am mostly convinced and want to try adaptive designs, and I see things differently now and will use adaptive designs. Again, the question is, after attending this presentation, how much more are you inclined to use these techniques? You're all much faster this time around. Thank you for your participation. Let's take a look at where you voted on. 47% saying you've still got some questions, but you're willing to try it. 37% you're more convinced now. 12% I see things differently. And then 5% you still feel the same as you did before the webinar. Once more, thank you for your participation. And once again, I'll pass it to Dr. Schnipp.
Thank you, Ryan. Well, it looks from the poll like at least we've allowed some people to open up their thinking process on the use of adaptive designs. And I hope what we have at least started people thinking about is that the use of these designs are not necessarily more complicated than more standard designs. The input to them on the point of the clinician is not more difficult than they would need to use in other designs. That the math may be complicated, but once the algorithm is set up, to use the algorithm to decide on doses within a trial is actually fairly straightforward and would actually allow fewer patients to be treated at perhaps low and ineffective doses, and also fewer patients to be treated at too high and toxic doses, exposing less patients to doses outside what would ultimately be perhaps the phase 2 dose. So that I think that at least I, for one, now use more, put more thinking into these adaptive designs and being open to discussing this with my statistician to see whether or not the particular program that I'm working on can benefit from these designs. And I do it now with less concern regarding the complexity of the underlying equations, knowing that the input is relatively straightforward and the ability to use the output from the algorithm is also relatively straightforward and easy. So with that, I guess we would just open it up to some questions.
Yes, well, thank you very much for that insightful presentation. I would like to invite the audience to continue sending their questions or comments right now using the questions window for this Q&A portion of the webinar. I've already received some questions though, so we'll get started with those. Our very first question for our panel here today asks, do you think adaptive designs are suitable for radiopharmaceuticals?
So I'll start with this. I think that adaptive designs are useful from a statistical perspective. They are useful for any experimental product, and you know, without understanding the actual intricacies of the radiopharmaceuticals, I don't see any reason why you would not consider them for this. You know, consider one of these designs for a radiopharmaceutical product. If I can ask any of my fellow panelists, John or Ed, would you like to jump in and add your perspective there?
Yeah, I'll jump in, and then please fill in if you agree or disagree. I feel the same way. There's no reason to think that whether it's radiopharmaceutical or antibiotic drug conjugate, oncology-based targeted therapy could not utilize any of these sorts of adaptive methodologies for their first early clinical trials, or even later on in terms of you know, expanded cohorts or you know, adaptive designs to recalculate sample sizes. So again, Mike, I'll just you know, reiterate essentially what we've all said is, you know, finding that trusted statistician, and then as the clinician and researcher really questioning and exploring other alternatives to the traditional approach. Ed, did you have anything else to add?
Right. I think the only area where I would imagine that these adaptive designs may be less useful is if the dose range that you're testing is very restricted by some factor that's unrelated to toxicity. Dynamic range of the PK or the PD, where you're only going to test 1 or 2 doses for reasons that are not related to toxicity. Other than that, I think these designs should be considered pretty much routinely.
Well, excellent. Thank you very much for that answer. Our next question would like to know, from a stats perspective, what would you use one of these methodologies over another one? Why would you use one or over another?
Well, statistically speaking, I think with statistical properties such as looking for an unbiased estimate of the maximum tolerated dose, or looking for a dose which has a smaller variance, where the estimation has a smaller variance, these newer methods are actually designed with those as constraints in the construction of the algorithms. The 3+3, it doesn't have those similar sort of constructs in its build, and so you are getting a dose which is closer to the objective of the study, which is to find the true or to find the MTD. And from a statistical perspective, you minimize that error between the true MTD and the one that you've estimated.
Thank Mike. Also, let me just add one other thing. I think we've been talking more from a perspective of a quote-unquote traditional U.S.-based drug development program, and that if we start talking about phase 1 programs based, let's say in Asia or potentially in Europe, I think that they're very well versed in terms of these alternative methodologies and adaptive trials. But if we're talking about phase 1 trials in, let's say, like China or Japan or that sort of thing, I can't really speak from experience how accepting they are with these other methodologies. I don't know, Ed or Mike, if you've had experience with early stage development in other countries utilizing these other methodologies.
Yeah, I think regulators in Europe are probably more likely to at least listen to the argument. I think regulators in the U.S. are becoming more comfortable with it. I think there's a body of evidence which they are now, which all regulators are accepting. So I think there will be some more wide use. And it was good to see in that last poll question that only 5% of the respondents said that they want to stay where they are, or stay where they are. I believe that's what I saw. So at least 95% of the folks are interested in learning something about this, if not necessarily going to jump at their next study to apply it. At least they're interested in learning.
Excellent, thank you. Our next question first states that this presentation comes from an oncology point of view. Their question is, do you think these techniques are well suited for other therapeutic areas?
I think so, and I think in the statistics literature, it's been applied to other therapeutic areas. I was looking at one recent paper where the first sentence actually said that they're applying them in osteoarthritis and rheumatology. So, so they are being applied in other diseases other than oncology. Oncology has always been, at least in my opinion, it's always been the place where we get to try new designs, because there is a real need to provide therapies to patients. And if there's anything that can be useful, let's do it quicker rather than go through tests that may put patients at undue risk.
Mike, I'll pose a question back to you around this, and Ed, please, also jump in. So I've also known, that there are other, let's say, patient-based trials. So, trials enrolling patients whereby you're looking at these sorts of, pain in particular. I think that they've used adaptive designs for many years now in pain trials, early pain trials. But what about healthy normal studies? So let's say like a primary care, so one of these, early stage phase 1 trials with healthy normal subjects that are enrolled. At first I thought, going through this and really doing the research, I thought, oh, we should only be in patients, but do you think that we could apply this to, let's say, your traditional healthy subjects as well?
Well, from a statistician's perspective, if the design is going to help answer the question and work towards the objective, then it can be used. And if that's in a healthy volunteer study, then from a stat perspective, there's no limit. There's nothing guiding that. Are there ethical considerations or clinical considerations that you have to give before you do that? Well, then certainly I think those would have to be discussed.
And clearly, the toxicity rate that you would accept, in a healthy volunteer trial would be different than what you would accept in a, phase 1 oncology trial. So that would obviously affect the design issues. You would clearly not allow 30% toxicity rate, in a healthy volunteer trial that you might allow in a oncology trial.
That's a great point, yeah.
Mm-hmm. Excellent, thank you. Our next question would like to know, could you please comment on how to define the optimal biological dose? For example, for immuno-oncology compounds such as MABs, not necessarily have to find the highest dose possible that causes the least harm.
I think
that obviously is a slightly different issue when we're talking about finding max, dose-limiting toxicities, and in many biologics that really isn't necessarily the endpoint of the trial. So I don't know if Mike has comments on that, but when you're not talking about toxicity as the endpoint, then you're talking about a slightly different issue.
Yeah, I think this is a different problem or a different question that we'd be happy to discuss it offline. But I certainly think that our objective here was to kind of show that these designs can be simple to use, just like the 3+3. But this question's probably a little more complicated than the scope of what we'd like to do here. We're happy to discuss it later, but I think let's take that and try to solve it later.
Yeah, and I think just real quick, Michael, I'll add to that. This is also applicable to where I was going in terms of within Japan. So a lot of the drug development in Japan is identifying that minimally effective dose versus maximally effective dose. So yeah, I think it's probably best take it offline. I think that's a whole nother, maybe for Xtalks, that's another conference that they could target.
Excellent, thank you. Our next question is simply curious to know in which scenarios are adaptive designs not suitable?
I think Ed mentioned a few earlier. If you want to mention them again, Ed?
Well, I mean, for me, one of the particular designs or clinical scenarios where adaptive designs would be less useful is if the drug that I'm investigating has a very limited dose range, or I'm only going to test, 1 or 2 doses because of other issues that are not related to toxicity, such as solubility or volume or receptor occupancy or other kinds of issues where I'm only going to test a couple of doses because of the nature of that particular drug. I have a trial I'm doing right now that I'm only going to test 3 doses for a variety of reasons with that particular drug. And so I don't think an adaptive design would help me that much because I'm only going to test those 3 doses. If one of those 3 doses doesn't work, that drug is not going forward.
That's perfect. Thank you very much. Our next question asks, first they state that difficulty remains on which design to use and the challenges of the biomarkers and/or defining DLTs. Do you have any additional thoughts to help guide the selection of a design?
I think the design you choose should take into consideration, the population that your experiment will cover, and if there are any reasons for potentially exposing patients to a dose that's not effective, well, certainly you want to use a design that's going to minimize the number of patients or number of patients that get an ineffective dose. So I think that you need to take into consideration, the population, treating the disease. I certainly think it would not be, it would be difficult to enroll a study if there was a perception that the dose was not going to help. And so the fewer patients you enroll on those kinds of doses, I think the better.
Yeah, Mike, I think where this plays a part also is within the pediatric oncology space too, right? Whereby you may need to be, in order to dose escalate, you have to be exposing more and more patients or a significant number of young, at-risk patients to where we know is going to be, ineffective doses, and getting to where you think the more effective dose is going to be, the quicker that you can get there, the more you're going to get buy-in, from your investigators or researchers, but also potentially, patients and caregivers and parents.
Wonderful. Thank you very much for that answer. Our next question would like to know then, do adaptive designs typically accelerate trials?
Well, I'll answer that. I'm not quite sure about the word typically, but I will answer that on the average and in the long run, that adaptive trials are quicker. So the expectation is that you will get your answer with fewer patients. If that's a good definition for typical, well, then the answer is yes.
And also, fewer patients would be treated at doses that are farther from the phase 2 dose. And that has other implications in terms of, getting a sense of efficacy of the drug early on, which, is helpful, particularly, if you're trying to make a successful program overall. But I think the notion that you're treating less people at doses that are farther from the ultimate dose that you choose is also, an important advantage.
Perfect, thank you. Our next question asks, what about PK assessment for drugs with high variability?
I have to defer to one of my clinical colleagues on this and the interpretation of PK results. Generally,
I'm not sure how to fit in the context of what we've been discussing. I mean, we've been discussing basically trials that are designed around sort of a toxicity element, and I'd have to defer a little bit to some of my colleagues on whether or not a PK analysis is really would follow the same rules.
All right, that's great.
I understand.
Sorry.
Yeah, I understand where the question is going. Yeah, and I guess you're going to have some idea in terms of the high variability going into this first-in-man or first-in-patient trial. I think the goal really is then, how much of that PK is going to drive the next dose or the dose selection into the phase 2, and you're still going to be able to implement all your PK assessments, even with the Bowen, I think it's Bowen design. I mean, you'll have less PK at your low doses, but you're still going to have the PK at really the key doses in around your efficacious dose or your range of doses in your next trial or phase 2 trial. I don't know, Ed, did you have anything else to add aside from that? Nope, it's not. Ryan, I think we're good for the next one.
Yeah, so for our next question, assuming no DLTs were observed up to the highest planned dose, is it worth using the adaptive design?
I think so, and I think I think I would then challenge the researchers. Do you want to go to a higher dose? I mean, if the objective here, as it has been in oncology for many years, to the MTD is as the maximum tolerated dose to look for where DLTs occur, and then decide that's where you want to dose. There's a perception that if nothing is, if you don't observe any DLTs, then your dose is still too low. So I would challenge the team, maybe we should look at a higher dose and continue with the design, continue with the study.
Well, sometimes the maximum dose that you can achieve is not always based on toxicity. Obviously, there are issues of formulation, and perhaps the formulation has something in it that you can't have more than a certain amount of, or you get into volume issues if it's an IV drug, or protein load issues, depending on what kind of the drug is. So, some of these drugs may have a maximum acceptable dose that's based on something other than toxicity. And in those cases, you may end up coming to the end of your dose escalation, without, toxicity as being the endpoint.
All right, fantastic.
So
Thank you.
Your decision there, Ed, I think would be to just use that highest dose that you've gotten to. Is that correct?
Yeah, I mean, like I said, there are some drugs that I've worked on where for example, the formulation contains a liposome or a protein or something where there's a certain limit to how much of that formulation you'd want to give somebody at a time, and that defines the upper dose, regardless of whether or not you have a toxicity, because you've gotten to the limits of your comfort with giving the drug itself or the formulation that it's in, and then you would stop at that dose.
Excellent.
I guess also along that same line, Ed, there could be a commercial cost of goods rationale as well.
There could be a
commercial reason beyond that too. Good point.
All right, for our next question, they would like to know how you, the members of our panel today, have personally seen adaptive design take a tangible difference in a study for patients or for sponsors.
Well, I have seen what we were speaking about earlier, and that is that that we've been able to escalate without having to go to 3 patients in a 3+3. So that's a tangible benefit. The next dose up was, was well tolerated as well.
Yeah, I'm same Mike there. In my experience with early stage development within the adaptive designs, essentially using, say, one of those 4 methodologies to really accelerate in terms of next dose levels within those early stage programs. Really, and seeing some significant time savings, and also, let's say, buy-in from the researchers or the principal investigators once you get to higher doses and potentially more efficacious doses.
And I would just mention, although it's not maybe a little bit out of the scope of this talk, using adaptive designs in phase 2 trials, where it's enabled me, for example, to very quickly get into combinations and other kinds of things like that in a more phase 2 setting, which have been very helpful to move the project along and get into more complicated combinations and other things much earlier than I would have in more traditional design.
All right, well, thank you very much, all of you, for those answers. However, we've reached the end of the question and answer portion of the webinar today. Now, if we couldn't attend to your questions, the team at ARG will follow up with you after the presentation. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from Xtalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it will help us to improve our webinars. Now, I'm about to send you a link in the chat box, and with this link, you'll be able to share this with your colleagues and view the recording of this event on this page. Now, please join us in thanking our speakers, Dr. Friend, Dr. Schipner, and Dr. Wisniewski, We hope that you all found the webinar informative. Have a great day, everyone, and thanks for coming.
Bye.
I know.